right, so let's not pick up where we left off on our last meeting. We were talking about the components of a muscle twitch, right? A single skeletal muscle with a single skeletal muscle cell with a single stimulus. We identified the role of the twitch, and then the muscle can contract, and then the muscle can relax. And then we transitioned to a really cool feature about skeletal muscles, is that individual muscle cell twitches can sum together. What did that mean? They're going to add together, right? You're like, sum. The muscle twitches, the contractions can sum together, which is something new to us so far. When a muscle contraction starts, relaxation is initiated. If there's a new stimulus, the muscle can then, right, from that same point, continue the force of contraction. So we can increase the force generated by muscle cells if there's a more frequent stimulus. We also identified that the summation can continue such that we actually kind of average out a maximal force generated. So there's no discernible real relaxation, so the tension by the whole organ is maintained consistently. Excuse me. What term did we call that? Tetanus, right? We call the tetanic contraction. And then we learned with a continuous, very frequent stimulus, the tetanic contractions can be sustained, or the term is called fused, where there is no discernible relaxation. It's just the muscle cell is just completely stimulated. It's maxed out, if you will. And you can give it more stimulus, but it's not going to generate more force. It's just completely maximized in the force which it can generate. So there's some variation in the duration of the force generated by the muscle, as far as the length of time, and then just the force generated. I guess maybe that's something I should point out here. A skeletal muscle cell can maintain that contraction for a period of time. If there's summation going on, it may last for a few seconds or longer, really. So we can sum them, and then it can extend for a period of time. It doesn't have to on, off kind of thing. Which makes sense if you use your skeletal muscles before, right? You could just stand in place and hold a contraction if you chose to do so. All right, so what questions do you have about reviewing the phases of a twitch or how these muscle twitches can be summed together? What was the latent period of a muscle twitch? What was the latent period?
Isn't that this long space contraction?
It is a space, but it's not the space between contractions. If it's between contractions, I would just say the word relaxation. When that excitation,
As I said, that's when all that stuff.
When coupling is happening.
all that stuff, all that excitation, all that excitation, contraction, coupling.
The rigor mortis.
Well, the cross bridge would actually be the contraction itself, but everything before that, the inflate potential, the action potential, the calcium flood, all that takes time. And we would measure that during that tiny little latent period. It takes a bit of time. In the upswing, though, of contraction, the cross bridge cycling is actually happening. They use those contraction. The latent period, the physical attributes of the muscle, okay, when the stimulus is received and there is no force being generated, occurs simultaneous, the excitation, contraction, coupling. So to clarify it maybe, hopefully, excitation, contraction, coupling are those intracellular events that we can't see, but we know a lot about, we've been talking about for a couple of weeks. The latent period is a measurable period of time. So that time that those events happen intracellular that we can't see, even though a stimulus has been applied, whether it be like electrical shock or the nervous system, that gap between stimulus and actual measurable force is called the latent period. So maybe to answer your question, they're simultaneous. One's intracellular, the other's something we can measure time, force. Does that make sense?
Yes.
Does that help or confuse?
So it's before the excitation coupling, right?
The latent period Okay, it's tiny here. Let me go back to the previous image. The latent period is this little yellow right here. That is the latent period. Because we're measuring over time the tension generated. The pink thing would be where the stimulus is applied. So somatic nerve releasing acetylcholine, or if you're in a lab, you apply like electric shock, right? As soon as the shock or neurotransmitters received, the muscle cell has to undergo excitation contraction coupling, all that intracellular event. That is happening during what we could physically record called the latent period. Additional clarification or questions? And then, after excitation contraction coupling, the muscle would actually do what? Contract, so we'd have that cross-bridge cycle going on. But that's happening, we look at the mechanics, but we could also measure the force generated. We could put a load on it and measure. Give someone a 10-pound weight and say, lift this. Right, whatever the case may be. And then calcium goes back into the SR and the muscle would relax. So the latent period is a physical measurement of time and force that happens while those intracellular electrical events are occurring. Are we a bit more comfortable with the latent period? Maybe? So we looked at a single twitch, the different phases, and how they can sum together. And then this is where we left off last time. We were talking, or didn't quite get into it, but fundamentally you should be aware that where there's a skeletal muscle, it can change length, and there's kind of an optimal length for moving a load. Okay. If I want to carry some water-filled buckets, let's just say I want to carry empty buckets. Okay. What's the best way for me to carry them? Would it be like doing this, like my arms are completely contracted, or stretching them, okay, or just kind of in the middle? Kind in the middle, right? Your muscle can stretch. Let's do this. Let's be silly. Let's stretch your arm. Try to hit your neighbor. Right, stretch. Okay, so let's focus on your chest, pectoralis, right, biceps, right. You know that you can stretch muscles, right? And you could still, right, put a pencil over and you could still move it in that stretch state, right? You could also, right, let's pretend we're the chin and butterflies, okay? You know that you could, right, shorten those muscle groups, right? Or you could do the same thing with your biceps, okay? But between that most stretched to the most contracted state, there's a kind of an optimal length where you're intermediate, where the amount of tension generated is maximal, okay? So thanks for playing along. And that is kind of plotted here, right, without looking at a particular muscle organ, plotting the relationship between length on the lower end, the shortest, right, state of the muscle, and on the far right end, the lengthiest part of the muscle. It's plotted against the amount of force or tension that that muscle organ, right, could generate. So sure, a skeletal muscle being stretched out could generate some force, but not near as much of it as the intermediate, and not even if you're really just over-shortening, right, the skeletal muscle. And you would see that kind of play out on the most short end of the skeletal muscle. Okay, so we have that optimal length in the middle of contraction where the muscle has its maximal amount of force that it could generate. Okay, so it's dependent. So a shorter versus a longer muscle are going to generate less force than this kind of intermediate state. Can y'all see that or appreciate that relationship? In this image, you have as well the relationship between this, the thin and thick filaments of the sarcomere and this overstretched. Thin and thick filaments can't interact as much as if it's overcompacted. They're already overlapping. They can't overlap more to generate more tension. In the intermediate, we have kind of optimal overlapping between the thin and the thick filaments to generate that maximal force that the cell and the organ can generate. Any questions? Another thing that we need to take into consideration is the use of these things called motor units. So let's define a motor unit. You've got your guided notes on page 7, the very bottom of page 7. A motor unit, we defined it in the lab a couple of weeks ago, but we didn't really kind of pull it out. So we're going to talk about it here now. Does anybody remember the definition of a motor unit from their handouts?
Nerve fibers control the control nerve.
Has something to do with a nerve. Has something to do with?
Fibers they control.
Very good, very good. A motor unit is a single motor nerve, right? So it's one nerve and however many muscle cells which it controls. So one nerve, and you can say X number of skeletal muscle cells would make up a motor unit.
X number of
Muscle cells. So again, a motor unit is how many nerve cells? One single, it's one nerve and lots of, right? Many, several different muscle cells. What that tells us is that not all motor units are the same size. Some parts of our body, the motor units may control, say, just 5 little muscle cells. In other parts, it may control 20 muscle cells. Right? Let's do that. Description. So what happens is if we need to move something, remember we're in the voluntary nervous system, if I know that I want to go pick up a backpack off the floor, right, my body needs to plan for getting enough muscles recruited to do that action so I don't fall over or whatever the case may be. This actually ties right back into chapter 9. You may recall when we did the brain dissection and we talked about the role of the frontal lobe and how it plans voluntary movement. Do you remember that? Make it feel good? Yes? Yeah. Remember how the frontal lobe, the somatic motor nerves control out of there? And so they are always planning. You don't have to cognate it. Your brain's always kind of calculating how much things weigh, what muscle cells will be utilized if you're trying to move something. We take it for granted, if you will. Okay? Pick up your bag. Don't even think about it. You just probably got a habit now. Run out of the house, grab your bag or whatever. But based on your experience, your body, your brain has to talk with the cerebellum and the spinal cord and your muscle cells to make sure the right number of motor units are activated, so your movements are fluid and you can move the load. So not all motor units are the same size. What that means is some motor units are small and some motor units are large. Smaller motor units would have fewer what? Muscle cells. And so the amount of force generated would be less. Organs in our body that tend to have smaller motor units are the ones for very precise movement. So like, for example, your eye, controlling your eyes, very precise. Controlling your fingertips needs to be very specific, right? So those would be smaller motor units. Fine motor skills. It's a good analogy or clarification. What about larger motor units? They would have more muscle cells, more force, what type of movements? Kind of move, do it. Large movements. So think about like your legs, your back, your shoulders, so your big muscle groups in your body are going to have large motor units. And if we need to do something, what can we do with our motor units? If I need to do a bigger movement, what can I do? You can sum them, add them. What would you use? Use multiple, right? So if I need to increase my workload, I can just add more motor units to do the job. Have you ever looked at something? Say you're helping your friend move, and you see a big box, and you kind of see it sagging, and then you go, it's your job to carry that big box, and you go to pick it up, you're getting all ready to grab it, right? You're agonizing about having to lift that heavy box, and you go pick it up, and you like almost fall over, so it's actually empty. You ever done that? Right, something like that? You're like, no, I'm cooler than that.
Yeah.
But if you see a big box, and you know there's something, you presume that there's something in there, right? You presume it's going to have a bigger box, going to have something probably heavier in it. Your brain, without even knowing what's in the box, is going to predict there's something heavy in there. It's going to recruit however many motor units that it needs. And if what you actually measure isn't what you predicted, then you might have those big kind of over movements. It's pretty cool how normally we don't do that on every movement that we do. Our body's pretty precise in recruiting the motor units. This proportion of the large versus the small motor units is summarized in figure 12.19. Smaller motor units, precise movement, larger motor units, larger movement. And as we identified, if we need to increase the output, our brain can recruit those, so we have a nice fluid movement. Question?
Oh, was that supposed to be right there?
Yeah, so I'm just pictorially talking about the relationship and the recruitment. So this is just a nice continuation, hopefully, of what we were just talking about. So yes, figure 1219 should be headed in the gray. Do you see it?
Oh, I don't see the figure, but I see the
Right.
title.
Yeah, I didn't want to be redundant in the pictures. So what we're taking away is if you need to increase your output, you can recruit more motor units, and those motor units that have more muscle cells associated with them generate greater force. Tension. Additional questions or clarification? All right. So if we look at any given muscle organ in your body, you know, we don't have to quantitate it. You know that not every muscle in your body generates the exact same amount of force. If you're trying to pinky wrestle somebody versus, right, do a big wrestling move, right? You know that the muscle in your arm is going to generate more force than the muscle in your pinky, right? So not all muscle organs generate the same amount of tension. And the reason, right, is multiple, right, reasoning why. One is just the muscle organ size itself. Not all muscle organs are the same. We can go to the extremes, the eye and the muscles of the leg, right? Tiny, large. You can look within the organ and see how many or few motor units are actually recruited in that organ. And then we go back to what we said, the size of the motor units. And then we go a look at the actual muscle organ itself. Just because a muscle cell is innervated doesn't mean that it's actually responding to the stimulus. So you got to take that into consideration as well as the target actually responding. And then within a muscle cell, how much tension is being generated. Right? You can tie that right into the number of myofibrils. Does a muscle cell have a lot of myofibrils in it? Or does it have very few? And so there's a lot of variations why any muscle organ might generate a range of tension compared to any other organ in your body. To maybe help clarify point number 5 is think about, have you ever had an injury where you had to wear a cast? Right? wear a cast or right I don't know, something where you've restricted, right? You're moving a sling or something like that. Has anybody ever been around somebody who's had to do that for a while? What happens even just after a couple of days? My strength goes down. My muscle cells haven't died. What's happened to my muscle cells? The term is called atrophy. They don't take a nap. That's called atrophy. And what's happening during atrophy? Right, so I'm not using it.
Well, they waste away.
And the term is called atrophy. What's wasting away?
Isn't that dying?
They do get nourishment. They're still alive. They're just not getting used. So if your muscle, well,
They become useless.
your body may deem it as such, but if I'm not using them, the myofibrils aren't being used. So your body's like, why spend all this energy to keep these proteins, all that all that stuff you guys loved last time we met? It takes a lot of energy to maintain and keep that. So we're not using it. Let's break down those myofibrils. So think about say an arm in a sling for a week, before and then after. My muscle cells are still there. They're still getting nourishment. They're just not being used. It's like they're atrophying. So the tension generated be the same before and after? Same person, same muscle group? No. Because later, right, the myofibral count has changed. So if that happens, could you gain that muscle activity back?
Yeah.
Sure. By
Memory.
using it. Exercise. Use it again.
So what's that mean? Muscles have memory.
Memory. Your memory is not stored in your muscles, but it's stored in your brain. Just when you keep repeating something, you form a memory, which we unfortunately didn't get to talk about in chapter 9. But brain, just repeating something, doing over and over. You've tied your shoes all of your life. The longer you tie your shoes, the more you don't even have to think about it. So just repeated action, right, forms memories in your brain. And so if you're curious about that, you can go to chapter 9, Terrence. Of course, right, every day I think I say, we're building all semester long, and here I could say the same thing, energetics. Skeletal muscles we know require a lot of ATP to bind to which part of our sarcomere? Which part of the sarcomere will ATP bind?
To the mid.
To what? On the what? The thick filament head. Remember, the thick filament head has the ATP binding site. Each thick filament head needs an ATP molecule to power. If you kind of quantitate the billions of little myosin heads that may be in a given cell, it takes a lot of ATP to contract. So if you look at the summary table, I'm going to look at the middle and the third column first, because this is what you're familiar with. In the middle column is what process? Glycolysis. Uses which molecule? Glucose. Lots of molecules, but we looked at glucose. Converts it into pyruvic acid to produce for us 2 molecules of ATP. You may remember the type of phosphorylation. Oh, your savior over there, right? Substrate level phosphorylation. Very good. So skeletal muscles can use glycolysis to produce ATP very quickly. A couple of molecules produced. We know that if aerobic conditions prevail, that pyruvic acid can be catabolized during which process? What would happen next after glycolysis? Krebs cycle and what happens after the Krebs cycle? Okay, the Krebs cycle intermediate step, but then the big one, the ATP producing one, is the electron transport chain. You can collectively just call it aerobic respiration, right? Oxidative phosphorylation. Produced for us another high 20s, low 30s ATP. So, sure, skeletal muscles have normal metabolic properties. They can do glycolysis, they can do the Krebs cycle, they can do the electron transport chain to produce a lot of ATP. So those second two columns come from chapter 3. But what's new to us is this first column. Our skeletal muscles can produce ATP even faster than glycolysis can by utilizing a molecule called creatine phosphate. It's abbreviated CP, and you can see the word here in this second row. Creatine phosphate. It almost tells right on itself. What is it going to do? Phosphorylate what? AD. It comes from creatine, and it phosphorylates ADP to make for us ATP. By what type of phosphorylation? Substrate. Very good. So it's just a quick substrate level phosphorylation from a molecule called creatine phosphate. An enzyme does it. It just transfers the phosphate from creatine phosphate over to ADP to form ATP. How many?
2. No, 1 ATP.
1 ATP. Because it's called creatine phosphate. It's not creatine di or tri. It's just creatine phosphate. It has how many phosphates to donate? 1. So you can quickly, under anaerobic conditions, it doesn't require any energy, oxygen, let me restate that. Just quick, fast, substrate level phosphorylation. From here to here. To this molecule called creatine phosphate. Produce ATP. And then creatine would have been what? What is the word when I said it releases a phosphate? Dephosphorylated. Very good. Dephosphorylated. So when you do some type of physical exercise, like you're sitting there and you're listening, and then every now and then you pick up your pen to take notes, you're probably using which molecule? From? Where's my ATP coming from? Probably, right, creatine phosphate. You're quickly just not really doing much. And you just get up out of your chair and walk. By the time you get to the door, you probably just used creatine phosphate. If you continue to exercise and move, let's say you want to go walk around the campus mall to go get a snack. Okay, so you walk over there. Then we'll probably do which process? All right, creatine phosphates run out. Now we're going to use glycolysis, and then we keep going, and then we're going to utilize, we're still walking, aerobic respiration. So creatine phosphate is that initial source of ATP. We need it fast to power that cross-bridge cycle. But if the work extends more than a minute, we can get into, or excuse me, not even more than a minute, within the first few seconds, then we could get into glycolysis, and then if the work continues beyond that minute or so, then you can get into the aerobic respiration. So again, our first source of ATP for skeletal muscle contraction is creatine phosphate, which substrate level phosphorylates ADP. Beyond the first movement, then our skeletal muscles can rely upon glycolysis, which produces not so much ATP, but it does, but primarily pyruvic acid, which can be further catabolized during that Krebs cycle at oxidative phosphorylation to give us a lot more ATP. So if you're trying to exercise and lose weight, what type of respiration do you prefer? Aerobic respiration. This is summarized for you in figure 1223, looking at if you're doing light exercise, so walking, going out for evening walk or something like that.
And that would be the glycolysis, right? The light
Uh,
exercise.
The light exercise, well, we're going to plot them. Creatine, when you first start exercising, you're going to first use creatine phosphates. Then we can rely upon, you guys identified glycolysis. And then if we need to extend, right, more than a couple of minutes, we can utilize oxidative phosphorylation. Okay, so you're looking at light to moderate exercise. This is how it goes kind of in sequence like we saw in the image. So that's during light exercise. Maybe you want to highlight it here. Metabolism changes as we increase, right, the workload, though. And I'll explain why in just a minute. Again, this is just aligning to our previous discussion. As we increase light exercise, the initial source of ATP comes from creatine phosphate. Then it can become dependent on glycolysis. And then if it continues, the light exercise continues, we can rely upon aerobic respiration. So it goes in sequence as we kind of predicted. However, if we increase our workload, I want you to make a note here about the intense exercise. If your exercise increases, so you're going to the gym and you're bench pressing, right, 3 times your body weight, right? You're doing like 10 reps. Okay? What's happening besides you're crazy strong? What's happening?
You're using more energy.
I'm going to need more energy.
Your muscles are going to be contracting and relaxing more.
My muscles are going to be contracting and maybe not even relaxing as much, right? They're going to be contracting. Muscles squeeze. What are they going to squeeze on in there, in the muscle organ? What are they? The muscle's shortening, generating tension. And what's in your organ? Within the organ, not within a cell. Right? So the whole organ, so step back a little bit. So you're still in the cell, so step back and look at the whole organ. The whole organ contracts and squeezes. What's in there that's getting squeezed? Blood. My blood flow is being squeezed. My workload is going up. My blood flow is going down, but I need more blood, right? I need more oxygen. So when you're intensely exercising, even though your oxygen content requirements go up, the actual content to the organ actually goes down. So can we rely upon the electron transport chain during intense, heavy exercise? No. Okay, we cannot. And so therefore, what would we rely upon? Glycolysis. Okay, so you'll still see that first source of creatine phosphate, okay? But during intense exercise, the bulk of your energy is going to come from, or ATP transfer is going to come from glycolysis. Okay? Oxidative phosphorylation will be working, but not maximally, because your blood flow is decreased because you're squeezing on the blood vessels in that muscle organ. So is intense exercise a good way to maybe free up some stored fat cells? Right? That's not intense exercise, but you're more light to moderate, where you're relying upon which type of metabolic process? Oxidative phosphorylation.
How would that free up fat cells?
What if you're doing intense exercise but
Go ahead, no.
what if your intense exercise isn't using muscles as much? I mean, if you're running
Yeah, so I would not categorize running as, okay, I'm not a runner, but if I were comparing running to lifting extreme weights, I would not put them in the same category. Because when you're running, yes, you're activating the skeletal muscles, yes, you're burning, basically utilizing stores from like a fat cells. You're also using glucose, but you're also utilizing your fats. And so when you're running, you may get a good workout from it, but it's not going to have that compression on the blood flow, I guess, is a better way to say it, as say powerlifting or something like that. And so I would, if I were to just kind of, I'm not an exercise physiologist either, so if I were to categorize it, I would say running gets adequate blood flow to the muscle organ, so that not only glucose can be used, but also for oxidative phosphorylation, but also fats can be metabolized, and that's why running and aerobics and those kinds of things are good for weight loss. Intense, like powerlifting or something like that, restricts the blood flow to the organs, and so somebody who does weightlifting is going to, in their power workout, they're going to rely upon glycolysis and creatine phosphate. That's why you'll see those people supplementing creatine before or after the exercise, because they're depleting those stores and the glucose. So someone who's doing that exercise. Yes, but somebody who's into that probably doesn't have a large fat storage anyway. If you're to stereotype somebody, I'm sure there's lots of people who have both. Fat and muscle. Big, strong muscles.
Those Olympic people that have to eat a lot, they show their diet.
Yeah. So what happens is, without getting into it too much, when you exercise, okay, so we talked about disuse atrophy, right? So you don't use the muscle, the myofibrils break down. But what if you do use a muscle organ a lot? So you're powerlifting all the time. Let's say you're running and power, you're like really into exercise. And you run all the time, you power lift all the time. Your skeletal muscles, the myofibrils will become what?
Stronger.
They'll stronger and larger. They will, I mean, the whole muscle will stretch, right? So that's the point of contraction. But if you increase the workload on a muscle organ, it's going to build more myofibrils. Okay? So the muscle cells would appear larger, right? So look bigger.
They say with all exercises and movements, if you do them repeatedly, the body somehow gets used to that.
Yeah.
It's not doing the same amount of work.
Yeah, if you're into exercising, there's kind of a plateau phase or something, so that's why people like to mix it up, like the example of the running and the weightlifting, and then you would mix up your power groups, like I do my legs on Wednesdays, my arms, or that's why you mix it up. And then even then you would change up, act like a trainer or something, like I go to the gym, but I don't. But yeah, so if you work out, you want to mix it up and then not do the same thing over and over. So it's kind of becomes futile. But it's still good to exercise no matter what. And the point of this is that depending on the type of exercise is going to dictate the metabolism within the cell. It changes on the type of exercise. When you do some type of exercise like running or powerlifting or whatever, once you get to that tired phase, what's happening is you're depleting your cells of any stored nutrients which it may have. We know that oxygen is being used, right, if you're doing aerobic respiration. Sometimes, my abbreviation here, LA, lactic acid. I think we may have briefly mentioned that in the first unit. If lactic acid builds up after glycolysis, we've got to convert that back to pyruvic acid, my abbreviation there, PA. If any glycogen in your skeletal muscles or liver, has been catabolized, you can rebuild those glycogen molecules. ATP, we know it's been used, so we need to bring it back at the substrate, set point. Creatine phosphate has got to be reformed. So after exercise, all of these molecules have to be put back to set point. Excessive things removed, storage things re-put back into storage. And if you know, after you do an exercise, let's say you went running, right? How far, Carrie? I'm going to presume you run. How far do you run? Look at you, good job. Just this morning. Good job, I'm envious that you do that. All right, so 3 miles, right? After the end of your 3 mile, were you just like fresh, like you just woke out of bed after the 3 miles? No. Right? Water loss, heat transfer, respiration after the exercise. Just finish your 3 miles, you're done. Respiration. What about your rate? Yes. Still faster, deeper, right, breaths? After she exercised, after she ran, when she was done exercising, she was still breathing quickly like she was exercising, if not even more deep, more rapid, right, breathing patterns, right? So think about if you're powerlifting or something like that in the gym, same thing. After you do the exercise, when you're not exercising anymore, your respirations are still elevated. And the reason why is all of these factors have to be restored. And so what your body is paying is what's called the oxygen debt. And so if you think about after you do the exercise and how your respiratory rate and depth is elevated, even though you're not exercising anymore, metabolically you're trying to replenish everything and get rid of the byproducts, if you will. And that's called the oxygen debt. Okay, that extra amount of oxygen that you need to take in after an exercise. Put everything back to what? That point. Do you have any other runners in here? I'm just curious. Anybody else go for a morning run? I'm impressed. It was raining too. It's just like super warm. Okay, all right, well. Still, there's a good excuse to not run. It's raining outside. I can't run inside. Good job. All right. Do it.
That would be not wanting to go to work this morning.
Yeah.
It's raining.
All right. So oxygen debt, what does that refer to?
Your body has to pay even after you quit the physical stuff. All these nutrients and everything have been depleted.
Yep, so all that extra oxygen to replace the depleted or low nutrients and get rid of excessives. So that's called oxygen debt. If we look at figure 1229, if we look at figure 1229, it helps, again, another not technically precise picture, but it gives us an idea of what's going on inside of the skeletal muscle. You recall our somatic nervous system controls the contractile activity, but we also have to monitor the we have to monitor it as well. So not only do you have motor nerves, but you also have sensory afferent nerves. And the nerves wrap around muscle cells, okay, to monitor whether a muscle cell is stretched, that middle length, or the contracted state. So they're wrapped around it. This ties into what we studied in the efferent nervous system. Remember we talked about proprioceptors? Remember those? The sense of balance? We took away our vision. We had to rely upon our proprioceptors, our sensory afferent nerves in our skeletal muscles to kind of see where we are. Remember the activity? Head back. Whoa. So our afferent nerves, shown here in the blue, teal color, monitor bundles of cells. And depending on, as I try to illustrate verbally, I don't know if I have a separate header section here. The state of the muscle cell is monitored. If it's stretched, contracted, or in intermediate length.
What are those lines at the bottom showing?
Action potential conduction. So more frequent action potential conduction, and then less frequent. I think that's what it should be labeled as. All right. So our afferent nervous system monitors our skeletal muscles based on whether the muscles relax or contracted. And you guys don't need to, I'm not going to, these terms are precise, but I want you to just acknowledge. Our sensory nerves monitor the status of our skeletal muscles. When they're contracted or relaxed, or when they get. When you go to anatomy, you may use those terminologies. The muscle status. Continually monitor muscle status. So we always want to be in set point, homeostasis, balance, etc. Sorry, you can put this, and hopefully you can visually tell. The more stretched, relaxed, and contracted state. It looks like that's going to transition us now to our smooth muscles. Before we move to smooth muscles, I want to show a very brief video published by the Discovery Channel.
This guy owes his strength to the muscles that move his bones. We all do. Take all water out of our systems, and the 656 different muscles in our bodies account for half our weight. By contrast, the bones that shape and support our bodies and give our muscles leverage weigh half as much. Our bones are light and strong. Inside, bones are a network of blood vessels and nerves. The centers of some bones are filled with marrow, which produces blood cells. Living bones grow and are replaced as parts have worn away. Calcium and phosphorus, the minerals that build the hard part of bone, gradually dissolve and are carried away by the bloodstream. New bone is built up from minerals supplied by blood vessels lacing the bone. Our backbones provide protection for the spinal cord, a highway of nerves carrying messages from the brain to the rest of the body, including all of our muscles. For every bone that moves, there is at least one set of muscles moving it. Joints, like the knees and elbows, allow our bony parts to work together and amplify the movement of our muscles. Here, the elbow enlarges the motion of the bicep muscle in the upper arm. This range of motion is accomplished with a mere 1 inch of contraction of the muscle. Not all well-built bodies need to look like this. Athletes can also be lean, training their bodies for endurance rather than short bursts of power. All muscles receive their instructions from the central nervous system. Electrical signals and nerve impulses travel to the spinal cord and then to the individual nerves in muscles. The impulses contract muscles, making them work. They're mounted on a flexible skeleton connected by joints. Taken all together, it's a human work of art in motion. This guy owes his strength to the muscles that move his body.
Okay, so we're going to now transition to the second of our 3 types of muscles, which is smooth muscle. I'm going to spend some time on this image, and you had a similar one in the handout that you're going to turn in on Thursday. So we'll review it. I'm trying to take some notes. If you recall, our 3 types of muscles, we had skeletal muscle, cardiac muscle, and smooth muscle. There's going to be some notable differences of our smooth muscle when contrasted to our skeletal muscle. The most obvious one is visually speaking. Your skeletal muscles are striated, whereas your smooth muscles are not striated. So you won't be able to see the thin and thick filaments with a light microscope in smooth muscle like you could in the skeletal muscle or in the cardiac muscle. They are the thin and thick filaments are there. They're just arranged so that they're not easily differentiated. Right? Where the thin overlaps the dark and whatever. So, and smooth muscles are muscles that can contract, they have thin and thick filaments, but they just are arranged differently. Okay. Anatomically, they're built differently. Control-wise, they're different. What type of control did we say about the smooth muscle? Autonomic and intrinsic. We have intrinsic and extrinsic control over some of our different types of smooth muscle cells in our body. And this image isn't maybe the best, but I like it because it kind of is concise in summarizing what's going on. Looks like I got it from the University of Wisconsin. In this image, let me just kind of set you up. Here in the middle we have the circulatory system, representing a circulatory system. On the margins, we have autonomic nerves. And did we talk about the varicoses in this class? In chapter 13, 12? Let me go back. Chapter 11, we said the autonomic nervous system has those yes, we did. I remember y'all are looking at me kind of like you're doing now, actually. We did talk about varicoses of the autonomic nervous system. So this tells me, I can see these things that tells me that's an autonomic nerve. And in the middle, in the gray area, are representing smooth muscle cells. Okay, so clearly our muscle cells don't look like this, but it'll give us an idea of how these cells work. On the far left, we have what's called multi-unit smooth muscle cells. So you may not be able to see it well, so write this larger if you need to. Multi-unit. And on the far left, we have single unit smooth muscle cells. They're both types of smooth muscle cells, but their control is different from one another. And their arrangement in the tissue is different from one another. So take a moment and just look at the smooth muscle picture between the multi-unit and the multi-unit and the single unit smooth muscle cells. What is different between the two types?
Oh, propagation.
Okay. Single unit cells are connected by gap junctions. So our single unit smooth muscle cells are anatomically connected by gap junctions. And what is the function of these gap junctions then? You're saying it. What's the function of the gap junction?
They're quick to pass. Very fast.
Electrical impulses, action potentials. Our gap junctions conduct action potentials, or let ions diffuse from one cell to another. So they're electrically connected, if you will. Do we see any gap junctions in the multi-unit cells? So they're not functionally connected to one another. What else besides our gap junctions can you pick out from here that's different?
We only have one varicosity.
Okay. We noticed that the varicosity relationship between varicosity, the number of cells controlled, is different. The single unit has, well, in this case it's one, so I'd say fewer varicosities controlling many muscle cells. By contrast, the multi-unit, the relationship between varicosity and cell is proportionate, right? So more varicosities control, let me say this properly. The multi-unit cells are more dependent on the nervous system.
Which one is more dependent?
The tissue type that is more dependent on the nervous system is the one that has more receptors and varicoses, which would be which? Multi-unit smooth muscle cells. What is the job of any cell? Any muscle cell's job is to movement and contraction, right? But something is unique here in the single unit in that some of our smooth muscle cells actually don't contract. What is their job? There's some muscle cells. Kinda. There are some smooth muscle cells whose job is to not contract. What is their job? Conduct. Initiate. What are we initiating? Action potentials. What are we going to call those types of cells? Not contract. They don't contract. We don't want to call them contractiles. That would be really, really confusing. Pacemaker cells. Very good. Your single unit smooth muscle cells have some set of cells called pacemaker cells, whose job is to spontaneously make and initiate and conduct action potentials. They regularly depolarize to what? Threshold. So your single unit smooth muscle cells have pacemaker cells in them that are connected to the rest of the cells. The rest of the cells, we can say they are what type of cells? Functionally, they are contractile. So in this image, in the single unit, top one, let's label that a pacemaker cell. We can see it is leaking a calcium is identified. It generates an action potential to, we should label the next two cells the what? Contractile cells. So a pacemaker cell controls the contractile activity of our contractile cells.
And so the pacemaker controls the contractile and the contractile and then
the job of the contractile cells is right there in the name. They contract. They generate force, generate tension. Where are we going?
I thought I read something else later. Maybe it didn't say
I'm not sure where you're going. Maybe you're going off where I said how the thin and thick filaments are arranged differently, but they are there in the contractile cells. There's contractile machinery in the contractile cells because their job is to contract. But in the single unit muscle, they can contract when they are intrinsically controlled by the pacemaker or even extrinsically controlled by the autonomic nervous system, or even controlled extrinsically how? What is this? What word did we say? Humorally. So sure, our single unit nervous system does have a source of intrinsic control, but we know it can be responsive to extrinsically by the autonomic nervous system and even humorally, right? Factors in the blood. By contrast, multi-unit cells, do they have a source of intrinsic control?
Which one, sir?
The multi-unit, do they have a source of intrinsic control? We got both coming in. The question was, do the multi-unit cells have a source of intrinsic control?
Yes.
I'm going to say no, because the intrinsic control comes from pacemaker cells. Do our multi-unit cells have intrinsic pacemakers? No. So our multi-unit cells are strictly dependent upon extrinsic control. predominantly from the autonomic nervous system, but certainly also responsive humoral. So we've compared and contrasted quite nicely a single unit smooth muscle type versus a multi-unit smooth muscle type. One, they're smooth muscles. Two, both types are controlled extrinsically by the autonomic nervous system and humorally as well. Where they differentiate is that single unit also has a source of intrinsic control from the role of the pacemaker cells. If we look at the effects though, when the single unit cells get excited, all contractile cells that are connected are going to contract together, right, in uniform type, if you will. So if this one unit has 20 cells connected, all 20 cells will excite together and contract together. Is that true for a multi-unit smooth muscle cell? They're all independent of one another. And multi-unit smooth muscle cells are independent of one another, completely relying upon extrinsic control.
So that's kind of due to the gap junction?
It's the single unit coordination is due to the gap junction connection. Is there a question or clarification? All right. Single unit smooth muscle, all of the cells of a single unit work together as a, what's the name tell us? As a single unit. Multi-unit cells, kind of like a free-for-all, but it's controlled right by the autonomic nervous system. What questions do you have at this point about the difference between the single unit type of smooth muscle versus multi-unit?
So on the single unit, is that like they all contractively start sometime, or it's like the wave? Like
It'll be like a wave because it's going to take a, but it's not going to be, it's not prolonged because those ions flood really quickly. But it would be,
and then it doesn't go quickly.
I can't answer that properly for you. The question was, does it go backwards? My gut response would say you're right, but I can't verify that because I don't know if there's a refractory period to prevent that. So I will have to look that up for you. Good question. Additional questions or clarification? So, right, good old action potentials coming up. I love them. All semester long. This is membrane spontaneous depolarization. Figure 1237 in your textbook. This is which type of cells would be spontaneously depolarizing? Which cells would spontaneously depolarize? The pacemaker cells. So put a big title on this. This is in your pacemaker cells. Your pacemaker cells are leaky to which ion did we see in the previous image? Calcium. They're leaky. This one messes with us. Our pacemaker cells depolarize when there's an influx of calcium. I have it labeled. I don't have it labeled for you, but this upswing, depolarization due to the influx of calcium. In which type of cells? Pacemaker cells. And Randy, this answers your question right here. Hyperpolarization is there, so we wouldn't see it going backwards. So because our pacemaker cells are permeable to calcium, really leaky to calcium, calcium is always flooding into the cell, initiating contraction. in the contractile cells. Steady. I have a couple of images. I didn't provide them to you. They're from some fairly recent research. This one looks like it's from 2010. Look, this is the time phase. Let me just focus on this first panel. This is a smooth muscle cell membrane, and then we have some calcium channels. And it shows us that calcium can leak into the cell. Can you see the calcium, the black dots flowing into the cytoplasm? They're leaking to the calcium. And if we look at within a skeletal muscle cell, not a skeletal muscle cell, I'm sorry, a contractile smooth muscle cell. Figure 1235 will walk us through the steps of excitation, contraction, coupling in a smooth muscle. Remember, we took a few days on the skeletal muscle. The series will be similar, but certainly not identical in the smooth muscle. The smooth muscle is built differently. And so some ion sources are what's variable. So when a smooth muscle cell gets excited, again, whether it be intrinsic or extrinsic, whether pacemaker cells telling it to do this or the nervous system's telling it to do whatever, a smooth muscle cell is going to become excited when there's an influx of which ion? Calcium. Calcium comes from where? My text is covering it up. Where's the calcium coming from? From here, but also from the extracellular fluid. What's different about our smooth muscles, the contractile cells, is that the calcium comes extracellular initially. So initially, calcium comes from the extracellular fluid into the intracellular fluid. Then the calcium will flow from the what? The smooth ER, endoplasmic reticulum, the smooth ER, and the sarcoplasm. If you really want to use the S, you can use the word sarco, because that almost is misleading, because it's skeletal muscle, but sarcoplasmic reticulum, smooth ER, whatever you want to call it, will leak its calcium after calcium comes from extracellular fluid. Whatever excites it, calcium is going to go from the extracellular fluid inside, which leaks calcium from the smooth ER also. Right? So we have two sources of calcium, extracellular and endoplasmic, smooth ER. Calcium is going to do something completely different in the smooth muscle and the contractile cells. So our smooth muscle cells are built differently. So we're going to go on a pathway. Okay, this, then that, then this. All right, you ready? We're going to do this in 2 minutes, and then we'll review it on Thursday. Okay. All right, so what happens is when calcium comes first from the extracellular fluid, then from the SR to the cytosol, it binds with a protein called calmodulin. It's a protein called calmodulin. Happens to regulate calcium. Extracellular fluid calcium into the cell, smooth ER calcium into the cytosol. That will bind to what? Calmodulin, which is like a messenger. So then we're going to go do something else. Calmodulin, complex with calcium, is shown in the purple little sphere, binds to an enzyme called myosin kinase. Calcium calmodulin binds in the green thing called something called myosin kinase, which happens to be an enzyme that, do y'all remember your kinases? What do? What do our kinases do? What the kinases, enzymes speed up reactions. Yes, kinases do a specific reaction. Phosphorylation. You're thinking of some immune. Kinases phosphorylate. Kinases speed up reactions. Yes, the reaction the kinases speed up are phosphorylation. Substrate level phosphorylation. So kinase is an enzyme that will phosphorylate myosin. Well, that's good, because our myosin needs. What does our myosin need to power? Energy from phosphates. Okay, so calmodulin activates myosin kinase, which then allows for. All right. Where's this picture? Okay. Don't write all this down. Calcium calmodulin activates what? Myosin kinase, which phosphorylates myosin, as the name implies, which gives it the ability to contract. So keep it on the right side. Calcium calmodulin will activate myosin light chain kinase, which phosphorylates myosin. To give it energy to contract. We'll conclude our discussion here. This is figure 1235 in your textbook. We'll pick up on Thursday the rest of this. But before you go,